you can see just a diagram of it down here. As it happens, these two genes are very close to each other on chromosome 11. And the analysis involves cutting this gene into tiny little pieces using very specific molecular scissors called restriction enzymes. And in the majority of people, you would end up with a fragment that's that long and a fragment that's that long, and you can separate them out on a gel, and this is what you get, this sort of fragment, the two pretty large fragments. In some individuals, there's a variation. So you get the same length here, but a shorter fragment here due to the fact there's been created in the base sequence, the nucleotide sequence, a variation called polymorphism, which results in an additional cutting site at this point here, and you end up with a short fragment. So these individuals, this individual here and this individual here, are heterozygous for that particular variant, and this individual here, quite unusual for that rare allele, for that rare variation in which that cutting site exists. And if you then look at the frequency of that particular polymorphism in patients with myocardial infarction, those without, you can see that the frequency is much greater of that variance in the post-myocardial infarction group. And as it happens, in a recent genome-wide association study, they identified precisely that locus that was associated with risk of the many that they looked at. And, you know, this was total luck. You know, my colleague and I thought, and actually the gene probe, that radioactive probe that was used in this study, was available. You know, we were attuned to that, but that was one of the first gene probes for an apolipoprotein gene.